Oh,
Hello, can you hear me?
yes, we can hear you. We can see you.
Okay, then let me start. Okay. Okay, welcome back.
Start your lecture.
Okay, welcome back. Today is the class for chapter 3. Today I would like to share my screen first. Okay. Okay, can you see the PPT? Okay, today's topic is new drug development for cancer therapy. This is the third class of our course. And here shows the outlines for today's class. First, I would like to introduce about tumor, and then I want to talk about the immune system and the tumor. I would like to introduce how the immune system to remove or to monitor our health against tumor. And then last, if our immune system failed, we will develop the cancer in our body. And then this time we need drugs to treat this cancer disease. So last, I would like to talk about the development of anti-cancer drugs. So this is the outlines for today's topic. First, let's talk about the introduction first. So first of all, I would like to introduce the concept tumor. Okay, tumor is an abnormal, benign, or malignant new growth of tissue. So most of the people, when they heard the doctor said you have a tumor, then they will think they will think very scary. However, not every tumor is very dangerous because it can be divided into two types. One is the benign tumor, and another one is the malignant tumor. So it's a little bit different. However, no matter benign or malignant, the tumor possesses no physiological function in our body, and because it can be because they proliferate very quickly, so usually they will become uncontrolled and then further do harm to our body. And for tumor, it is also called neural growth or neuroplasms. And as I mentioned before, based on if it's dangerous or not, it can be divided into the benign neuroplasms or the malignant neuroplasms. The later one, we can call it cancer, which is dangerous to our body. And this slide shows the induction of tumor. As you can see in this slide, what can cause tumor formation? First, as we can live longer and longer now, sometimes the tumor can be induced spontaneously. As we get older, we have a higher chance to induce tumor in our cells. So it will cause the high rate for tumor formation. And also for other conditions, for example, the UV or ionizing radiation can also cause tumor formation. As you can see, this figure, this is a figure represents there is a radiation inside the room. Sometimes some of the immunological labs will have this local sign on top of the door. If you see this the sign on the door, then pay attention. Maybe there is radiation inside the room, so you need to get rid of it, because high dose of UV or ionizing radiation can cause gene mutation and then cause tumor in humans. And next, another factor to induce tumor would be their genetic abnormality. What's that mean? If you read many case reports or as an intern or doctor in the hospital, you will find that many cancer patients have the family history. That's because in their gene, there are some oncogenes inside their body, and which cause them have a high chance to develop tumor when they get older. So these genetic abnormalities can also increase their development of tumor. And next is the condition of our immune system. As we know that we we have an immune system inside our body which can recognize the tumor at the very beginning. If we have a good condition of immune system, they can find out the tumor cells at the very beginning and then kill the cells so that protect us against to induce tumor in our body. However, if we have a bad lifestyle, for example, we smoke, we have a very bad living habit, for example, stay late at night, drink a lot of alcohol, or our environment have very pollution, all this can cause another chemical carcinogens inside our body. And when the tumor cells overcome our immune system, and then the cancer cells can proliferate, and the tumor size will get bigger and bigger, and then will defeat our health. And another factor would be the hormone. That's can be found in some of the cases. For example, the obese woman, if they have a high level of hormone, they will have a higher chance to develop breast cancer. So this time, this hormone levels may also induce a tumor formation in some of the patients. And last would be the virus-induced tumor. So this one, I have a further explanation in this slide. As you can see that account for the studies, about 12% of the worldwide cancer incidence is attributed to the infections. For example, in this table, this EB virus can cause the cancers, and also the HPV. I think this virus is very famous. Human papilloma virus can cause the cancers in women, and also the hepatitis B virus or the hepatitis C virus can also cause the liver cancers in some of the cases. So so many infection related to the cancer formation. So what's the mechanism for that? Here shows some of the potential explanations. For example, all these virus, when they infected the hosts, they will try to create conditions for their replication. Then they will induce the cell cycle, they will change the metabolic reprogramming, and finally, all this change to the local tissue or local organ can induce a lot of angiogenesis, more vessels there, and then make the gene mutation finally in the host, which can cause cancer development. So to this point, if we can control the infection of this cancer-related virus infection, then maybe we can prevent the formation of tumor. So this is one idea to prevent the development of cancer maybe. So nowadays, already some of the labs or some of the hospitals can already develop some of the vaccines to protect the patients against this virus infection. So with these vaccines, maybe they can reduce the formation of tumor in the future. And next, this slide shows the tumor microenvironment. This is a very new concept, which is stated fewer than 10 years. So at the very beginning, a lot of the treatment for the cancer, they will use a lot of cytotoxic agent to kill the cancer cells. However, most of the cytotoxic drugs will get less effective, and they will develop the drug resistance later. And then when the scientists look more details inside the tumor tissues, they find that not only the cancer cells in this microenvironment, but also a lot of other cells. As you can see in these figures, this during the carcinogenesis stage, there's a lot of cells in the local tissue, including the stromal cells, the immune cells, and all these different cells, they can secrete a lot of factors or cytokines and growth factors, a lot of things. So inside this environment is very complicated. And when they have a higher level of uncontrolled factors secreted to stimulate the local tissue, and some of the, for example, the somatic stem cells or somatic progenitor cells or somatic differentiated cells, some of them may, under this stimulation by these secreted factors, they get mutation. Some of them may develop to this CSC, cancer stem cell. So these cells, they have the characteristic of their stem cells, and they don't have the normal function of the cells. So this cancer stem cells can get self-renew to develop more cancer stem cells, and some of them they can get differentiation, and then they get a tumor brick. So inside this tumor brick, still it's a complicated cell mass with a lot of different cells. So during this stage, it changed from the carcinogenesis stage to the tumorigenesis stage. So the tumor is developed here. And also our immune cells fail to kill all of this tumor brick. And also the stem cells, cancer stem cells are usually not response to most of the cancer drugs developed in the clinic. So this cancer stem cells get proliferated, and then after they get enough number, they can metastatic to other tissue. For example, if at the very beginning, if this cancer is developed in only lung, then through the vessels, they may move to the kidney, or they may move to the bone or something, some other tissue else. So then we'll get the metastasis of the tumor, and then a lot of organs developed this cancer, and finally the patient will die. So why nowadays most of the agents cannot control their tumor development very well? Some of the scientists think maybe due to this cancer stem cell, because they are not respond very well to most of the anti-cancer agents, and they have a high proliferation rate, and they can renew very quickly. So one of the opinions said, if we want to cure cancer in the future, we need to figure out a way to kill this cancer stem cell first. So this also a very hot study field for the anti-cancer drug development. So this is the concept for tumor microenvironment. For this tumor microenvironment, one point you need to remember: inside the tumor tissue, not only we have the cancer cells, but also a lot of other cells. So when you try to develop new therapy or new drugs to treat cancer, you need to figure out how to deal with all these cells so that you can increase the effectiveness of the new therapy. And after that, I want to introduce the immune system, how the immune system to kill the cancer in our body. For this one, we can introduce one of the science we call tumor immunology. So this is a very new science focused on the immune system and the tumor, the relationship of these two things, how our immune system find tumor cells, how to kill the tumor cells, and then the tumor cells, how to escape from this monitor of the immune system. So it's a balance. Once the immune system fails, the tumor wins, and then the patient will die. And as I mentioned, it's not a very long history science, but inside during this about less than 100 years, still have some hallmark for the development of tumor immunology. The first hallmark would be the concept about tumor antigen, which is stated by this scientist, Prof. He thinks that there may be tumor immunity. He thinks that there is a high frequency of aberrant germs, which now we know these aberrant germs are tumor, which if this tumor not kept in check by the immune system, the tumor will overwhelm us. That means that our body will have an immune system to keep our safe against the tumor. If this immune system fails, then we will die. That's the first finding. He thinks that's because this tumor has tumor antigen, which is different from our other things in our body. So the immune system can recognize it's the aberrant cells. We need to kill them. So this tumor antigen is the first hallmark for the development of tumor immunology. And also this tumor antigen is very important concept for all of the cancer drug development. And then the next hallmark too is about the immunosurveillance, which is stated by these two men. One is the Burnett, another one is Levis. So this immunosurveillance is a term used to describe the processes by which cells of the immune system look for and recognize foreign pathogens. It could be the bacteria or viruses, because the infection is highly related to the development of tumor. So our immune system, if they can find these foreign pathogens and kill them or remove them, we can prevent the formation of tumor. And also, if our body already developed the cancer cells, our immune system can also clear these precancerous and cancerous cells in the body. So it's the immunosurveillance to keep the health of our body. So as you can see, these two slides. This shows the brown dots. They are the immunocells staying with some of the antibodies. So if we developed aberrant cells, if aberrant cancerous cells in this tissue, a lot of immune cells will migrate and get into this location, and then they try to clear the cancer cells. So this is the evidence to prove the immunosurveillance. This is the hallmark second two. And then the last hallmark, which is important, is the phases of immunoediting, the 3 E's. So what are the 3 E's? The first E is elimination. So what's elimination? What does that mean? As you can see in this figure, there's a lot of red cells, right? Those red cells are tumor cells. So due to different reasons, it can be virus infection, it could be mutation or the genetic abnormality from the family. Either factor, either reason, then cause mutation and tumor cells formation in the local tissue. And then, as I mentioned, we have the immunosurveillance, right? So at this time, a lot of immunocells will come or migrate to this local tissue, including different types of the immune cells. As this one, the brown one is the macrophage, and the NK is the natural killer cells. And also the T cells, different types of T cells, including the CD4 positive T cells, CD8 positive T cells, the natural killer T cells, or the alpha delta T cells. Then all of these immunocells work together, and then they can clear or they can kill all the mutation cancer cells. Then they can protect our body. So this one is the first E, elimination, means the immunocells kill the cancer cells, and then we keep safe and we keep healthy, right? However, sometimes if the immune cells or immune system are not strong enough, then the tumor cells survive, and then later they would change to another balance, which is called equilibrium, which means that the tumor cells can survive and the immune cells are here. The cancer cells will cannot proliferate a lot or develop a lot because they have a balance between the new immune cells and the cancer cells. They can survive, but they cannot have too many in the local. And then they have a balance, and this is the second E. And the last one, when the tumor cells get more and more, and they can develop other techniques to escape from the immunosurveillance of the immune cells. And this time, the cancer started to out of control, and then they can migrate from the local formation tissue to other organs. So in this type, if we find the tumor very late stage in the escape stage, usually the prognosis of the patient will very poor. They have a low 5-year survival rate. So this is the 3 E's. First, immune cells kill tumor cells, and then they can get together well, and then the tumor cells win and the patient die. So this is 3E. So if we can find a tumor at the very early stage, for example, in the elimination stage or the equilibrium stage, then the outcomes of the therapy would be good. However, if in the later escape stage, the outcome would be poor. Okay, this is the introduction about the thesis of immunoediting. And here, go back to this concept, tumor antigen. As I mentioned, because the tumor antigen, this concept is very important. So I would like to talk a little more about it. So what are tumor antigens? Tumor antigens are abnormal substance produced by cancerous cells. So they are the product from the tumor cells. The body of the host means the patient with the cancer can react to this antigen, recognizing them as abnormal. So briefly, you can think that tumor antigens is something produced by the cancer cells, and our body can recognize that it's not from normal cells, it's from barren cells. So this kind of substance we call tumor antigens. And the classification of the tumor antigens, first is the tumor-specific antigen. Also sometimes say TSA for the abbreviation for the tumor-specific antigen. And it is also called tumor-specific transplantation antigen, TSTA, or tumor rejection antigen, TRA. So TSA, TSTA, TRA means the same. Means antigens only expressed in cancer cells, but not in normal cells. So only can be found in cancer cells, but not in the normal cells. Those kind of antigens we call tumor-specific antigen. This tumor-specific antigen usually they are controlled by the encoded mutation genes inside the cancerous cells. They comes from mutations. That's the reason why only present in the cancer cells, but not in the normal cells, because they arise from mutation. So only the mutated cancer cells have these genes. So they only can be found in the cancer cells. And they are usually unique, only found in the cancers. And the unique antigens can promote powerful immune responses. I think it's easy to understand because these antigens cannot be found in our body, normal cells, only in the cancer cells. So for the immune system, they can recognize it's the outside the things invasion in our body, so it can promote powerful immune responses of this tumor-specific antigen. And most tumor-specific antigen affect the intracellular proteins, which means that they're located inside the cells, and most of them are proteins. So after this patient get their chemotherapy or they get their radiation therapy, then the cancer tissue get injury. This intracellular tumor-specific antigens will release from the tissue so that they can be recognized by the T cells as the mutant peptide MHC complex on the surface of viable cancer cells. So this is how this tumor-specific antigen be recognized by T cells, because they're located inside the cells. So when there's cancer cells get injury, they can release this antigen, and then T cells can recognize it and promote powerful immune systems. But very few of the tumor-specific antigens also affect the surface proteins. For example, the mutant epidermal growth factor receptor, this EGFR, if you remember yesterday's class, you will remember this EGFR is highly expressed in a lot of cancers, especially for the lung cancer, right? So this kind of mutation EGFR belongs to one kind of surface proteins. So this also can promote powerful immune responses in our body. So this is the first type of tumor antigen, TSA, tumor-specific antigen. Compared to this tumor-specific antigen, another one is tumor-associated antigen. We call it TAA for abbreviation. So this kind of antigen, they express in both tumor or normal cells. So both the tumor and our normal cells can express this tumor-associated antigen. However, the expression level is much higher in cancer cells, which means that in the cancer cells, this TAA has a higher level of expression compared to the normal cells. For example, they could be the overexpressed molecules, for example, the growth factors and their receptors. Here shows some examples, the surveillance P53 and so on. And also they can be their differentiation or the lineage specific molecules, which means that some antigens expressed on the tumor cells are also expressed during at least some stage of the differentiation on non-malignant cells of the lineage from which tumor developed, which means that some of the antigens expressed in the tumor cells, they can also express in the normal cells in some stage during the differentiation of the normal cells. This kind of tumor-associated antigen, including CD20, CD19, they all belongs to this kind of TAA. And this lineage-specific antigens represents a very diverse group of proteins. Not only the protein, but also the carbohydrate, the glycolipids, they can also be some kind of TAA. However, the normal cell types that express these antigens will determine the toxicity and usefulness of any antibody or agent formed which are the target. What does that mean? That means if we use the TAA, the tumor-associated antigen, as the drug target to design anti-tumor drugs, because this TAA also expressed in the normal cells, at least at some stage, right? So for those patients, this target to TAA, the anti-cancer drugs, can not only kill their cancer cells, but also kill the normal cells which express the same TAA. So this kind of drug target may cause side effects. They will cause the non-specific killing of normal cells. So it will cause toxicity in clinic. So this table shows the comparison between the TSA and TAA. For tumor-specific antigen, they only express in tumor cells. So they have, if we use the TSA as their drug target, it has a high specificity because only cancer cells express TSA, but not normal cells. And also it can induce strong immunoresponse. This TSA was their origin. They can comes from the physical and chemical agents. It can cause by infection of viruses or other factors cause mutation. And the TAA, on the other hand, they not only express in tumor cells, but also in normal cells. Thus, the specificity, if we use this TAA as the drug target, they have a lower specificity compared to their TSA. And also because their normal cells also express this TAA, so it can promote weaker immunoresponse compared to TSA. And this TAA can comes from the mutant cell genes and come from the oncofetal antigens and differential antigens. So this shows the differences between TSA and TAA. So if you are thinking, use the tumor antigen to develop the anti-cancer drugs, I would suggest TSA would be a better target compared to TAA because more specific, lower toxicology in our body. Okay, then for next, the important part of our class today is talking about the development of anti-cancer drugs. So we already know what's the concept for tumor and the differences between the benign and the malignant tumor. And then we know that our body will have the immune system to protect us. But sometimes when the tumor overwhelms the immune system, and then the cancer is out of control, this time we need therapies to kill the cancer cells. So here shows the stages for the cancer drug development. First, on top of this slide is the preclinical development. So this one, similar to the medical research introduction we mentioned in the first class. Here we can design in vitro or in vivo experiments to get their potential candidate for further development of the anti-cancer drugs. And then for this, either in vitro or in vivo experiments, we need to design the toxicity and the therapeutic effects of the candidates and determine their safe starting dose for clinical phase 1 trials. So all this data we need to get from the clinical development. So we have a good potential candidate, and then we can do the further work. Then we can move to the clinical development. So in the clinical development, I think the students major in pharmacy know that there is 4 clinical trials for development of new drugs, including phase 1 trial, phase 2 trial, phase 3 trial. So if the candidate pass all the 3 clinical trials, they can get the license to be used in clinic. And further, with more and more patients use this new drug, and then later can start the phase 4 trial to determine any other uncommon side effects, assess the long-term toxicity or mobility of the use of this drug. So this is the normal cycle for the drug development. However, here, as you can see from the left, there is a phase 0 trial, and this is a new concept. For this phase 0 trial, low dose or short duration of treatment will be done in this stage, and then the company will have a small number of patients, and that the idea if this drug is good to perform phase 1 trial or not. So this kind of phase 0 trial, like the pre-trial before the phase 1, that's used for reduce the money cost for the company to have a failed candidate for the further work. So this figure shows the stage or cycle for the development of cancer drugs. We will introduce a little bit in the following slides. First, phase 1 trials. Okay, phase 1 trials represent the first administration of a new drug or drug combination to human beings. So this is the first stage for a clinical trial of the candidate. However, the oncology phase 1 trials a little bit differ from the non-oncology phase 1 trials. Usually the non-oncology phase 1 trials, they will recruit the healthy people as their volunteers to perform this phase 1 trials. However, the oncology phase 1 trials, they will prefer to use the cancer patients rather than the healthy volunteers. Why? That's because most of the anti-cancer drugs have a very level therapeutic index of the cytotoxic agents, which means that the side effect of the anti-cancer drugs is usually higher than other drugs. So they prefer to recruit the cancer patients rather than the healthy volunteers as their candidate for the phase 1 trials. That's a little bit different from other phase 1 trials. And most of the time, this oncology phase 1 trials are also offered to heavily pretreated patients. Those patients have no other therapeutic options, so they can be recruited for the volunteers for this oncology phase 1 trials because they have no other choice. So sometimes they can be recruited for the oncology phase 1 trials. Therefore, the selection of patients for phase 1 trials is critical. So you can see what's the standard for including these patients. First, patients are required to have good performance status. They cannot be very heavily ill, and they need to have the adequate organ function. They have a good function, for example, in the kidney, good function in the liver, which can help to minimize the chance of clinical deterioration from progressive disease that will require premature study withdrawal. So which means that we need to select those patients in relatively good condition, because when they use this new candidates, they will stop the old therapy they use sometimes, and then they try a new one, and that may cause very dangerous factors like the progressive of this disease and make this condition worse. So at the beginning, although they would choose the cancer patient, but they would try to choose those in good condition. And also, patients should not require receiving medications that may potentially interact with the experimental drug, which means that if the cancer patients undergo some of the therapy, then we need to make sure that those therapies will not interact with the current testing candidate. That's to avoid exacerbating the toxicity of the experimental drug or altering its pharmacokinetics. So we will have a very strict critical standard to selection the cancer patients for this phase 1 trials. Okay, The primary goal of phase 1 cancer trials is to collect data on toxicity, pharmacology, and pharmacodynamic properties of a specific drug or therapy regimen, allowing determination of the recommended phase 2 dose. So for the phase 1 stage, maybe the pharmacological effects not that important. During this stage, they make sure that this candidate is safe for the patient. So they may focus on the determination of the toxicity and also the pharmacodynamic properties. Then, due to the data from the phase 1 trials, the doctors or the pharmacists may have an idea what's the best recommended dose for phase 2 stage. And also, the drug interactions are also investigated in combination regimen phase 1 trials. Because, as we mentioned, most of the cancer patients undergo some of the treatment. So sometimes we need to figure out if the combination of the old therapy and the new candidate, are they going to have the drug interaction? So this question can also be answered from the phase 1 trials of the anti-cancer drugs. And then if the candidate passed their phase 1 trials, they can move to the phase 2 trials. The phase 2 trials is screening studies aiming to identify signals of anti-cancer activity in a homogeneous population of patients with a particular tumor type. What does that mean? That means that the patients for the phase 2 trials, they need to have the same particular tumor type. For example, they all have the grade 1 or grade 2 gastric cancer. And also those patients need to come from the same population. If we are doing the experiments in China, so they all should come from the Chinese people. We cannot match the American people and the Chinese people for the phase 2 clinical trials. We need to recruit their patients from the same population. And also, it is a critical step in drug development for this phase 2 trials, because a positive phase 2 trial can lead to a large or a more expensive phase 3 trial. So this time, some of their maybe more than 1/3 of their candidates may fail in the phase 2 trials. Okay, the primary goal of the anti-cancer treatment for the phase 2 trial is to improve the overall survival or improve the quality of the life in the patient. However, when we're choosing the endpoint of the study, sometimes it's difficult to use the overall survival, because as you know, from the patients get treated of the therapy to they die, it may take a long time. So due to the limited length of the observation and the confounding effects of subsequent therapies, and using overall survival is not that popular. Other endpoints should be chosen. So for the phase 2 clinical trial, usually they will choose the tumor shrinkage, which means that the candidate or the compound can reduce the size of tumor. That means the tumor shrinkage. This is a very commonly used endpoint in phase 2 trials. Another one is the progression-free survival. Also, is a commonly used endpoint in phase 2 trials. So for first, the tumor shrinkage, that's easy to understand when you measure the size of the tumor with different machines. And then you can figure out after the treatment, is the tumor size get smaller or not. And then you can have your evaluation for this effective of the drugs. And for another endpoint, for the another endpoint, not only consider the tumor shrinkage, but also we monitor the tumor stabilization in its definition. So you can think this PFS is a little more complicated compared to the tumor shrinkage, because they need to evaluate if any progression of the tumor when after treatment, is that any better? Are they stopped, or are they getting back, or are they developed in the tumor tissue? So this one is more, for me, I think it's more accurate. However, the main limitation of this PFS is that you need to frequently check the tumor, and you need to have a standardized assessment to evaluate the condition of the tumor. So it's a little bit complicated when compared to the only measure the tumor shrinkage. Then, if the candidate is good enough to pass the phase 2 trials, then you make to the phase 3 trials. Then these phase 3 trials are a critical step before licensing and consists of the benchmarking the efficacy of a new therapy against the best current standard of therapy. So what does that mean? So during this phase 3 trials, sometimes the pharmacist or the doctor will use a standard use positive control to compare with the candidate. Now you want to get licensing. So then you are evaluating two kinds of drugs. One is already in use in the clinic as the positive control. Another one is the candidate went through the phase 1, phase 2, now phase 3. This is the potential candidate. Then when you compare the effective of these two therapy or two agents, so what kind of medical research you are going to use for the evaluation? I think the best match medical methods would be the randomized control study. So in this point, you can recruit a group of patients and then randomize those patients into several groups. One group receive the positive control, the anti-cancer drugs already in use in clinic, and then the second group you can use this new candidate, and then you can compare the outcomes of each groups because it's randomized control study, and then you can have an evaluation to say if the new drug is better than the old one. That's the phase 3 trials suggest to use the randomized control study, as we mentioned here. Also, in most oncology phase 3 trials, the outcome is a time to event variable, such as we got the progression factor screening or overall survival. Sometimes the quality of life can also be a primary endpoint, because the improvement in the quality of life for the patient still be valuable in clinic. So sometimes quality of life can also use as an endpoint for this stage 3 trials. But using the quality of life is much less common than use of the survival or the primary as the primary endpoint. Okay. Then a new drug or drug combination might be approved if this rigorous conducted phase 3 trials met the clinical relevant primary endpoint, and then they will get licensed, and then they can be used in clinic. After that, maybe the phase 4 post-marketing trials can be conducted, but unfortunately, it's not that common. But if they perform the phase 4 clinical trial, they usually focus on the long-term safety or the long-term mobility of the treatment. But this kind of clinical study is not that common because it costs a long time and also a lot of money. So they are not frequently done in clinic. Okay, this is the table summarize different stage for the anti-cancer drug development. And as you can see, there's two types of different anti-cancer agents. The first one is cytotoxic agents. I think this is the all types of the agents used for cancer therapy. As say in this concept, it calls cytotoxic or it causes cytotoxicity inside the cancer cells, which means that they are toxic to cancer cells and then to cause cancer cell die or apoptosis or something else, and then they can treat the cancer. So this kind of cytotoxic agents. Another new concept or modern anti-cancer drugs is the molecular targeting agents. As said in this concept, they have a specific molecular target for those agents, which means that they can specifically recognize their target inside the cancer cells and specific kill the cancer cells. So these two different kinds of agents, I think the second one maybe have low side effects because they can recognize cancer cells. For the cytotoxic agents, they not only toxic to the cancer cells, but sometimes also toxic to the normal cells. So the modern anti-cancer drugs are more focused on finding new molecular target of the cancer therapy, and then they can reduce the side effects in the treatment of cancer. And both of the agents need to undergo the preclinical development, phase 1, phase 2, and phase 3. If they are good enough after the phase 3 clinical trials, they can get licensed and then sell in the market. However, the phase 4 is not required for every experiment. And here, as you can see, there is a phase 0 clinical trial, which is a relatively new concept in the anti-cancer drug development. So as you can see, this phase 0 trials focus on the molecularly targeting agents, which is more commonly used when you developed the molecularly targeting agents, because they can be helpful to bring a proof of mechanism of action in human beings. That means during the phase 0 study, the scientists or the pharmacists will confirm that the drug target inside the patient so that they can get confirmation of the action mechanisms of the drug. So this phase 0 is more related to the modern anti-cancer therapy development. Okay. And why they need phase 0 trials for this modern anti-cancer therapies? Because there are emergency of the molecular targeting agents in oncology not only have revolutionized the care of cancer patients because they are more specific, so they take better care of the cancer patients, but also they change the daily practice of the medical oncologists. These molecularly targeting agents often differ from traditional cytotoxic agents by their administration schedules and routes. So they may have a different therapeutic schedules, and they have a different routines and different toxicity profiles. So this time they need the phase 0 trials to confirm those information. And although it's a nice concept, but it's now well studied in many of the company. Most of the studies we can divided the phase 0 trials into 3 different types. The first is phase 0 microdose studies. So during these microdose studies, usually focus on the pharmacokinetics or the imaging. This kind of study are designed to evaluate the pharmacokinetics, metabolisms, and imaging distributions of specific antigens in the absence of any planned pharmacological effects. So during this phase 0 microdose study, small focus on after taking the small dose of the phase 0 child, how this drug absorb, how they metabolize, and how they distribute it inside the body. So they performed more pharmacokinetic data rather than the pharmacological data. And then the second type of the phase 0 trials has a pharmacodynamic endpoint. So these trials are designed to examine the pharmacological effects of the drug under investigation. And last, the third type of phase 0 trials is designed to evaluate the specific mechanism of action of the novel agent. That means that they will focus on find out the drug target inside the patient of these candidates. So they want to confirm this molecular targeting agents, if they are working the same inside their patients to target to the same drug target. So this is the confirmation of the drug target in human beings. So this is the third type of phase 0 trials. So as I mentioned, this phase 0 trials not that common or not required by all the studies. But nowadays, some of the companies prefer to do this phase 0 studies before they start phase 1, because the data from the phase 0 trials can give them an idea if this candidate is valuable or not to go further. Then they can save a lot of money if this candidate fail. So they can stop at a very early stage, so that they can save money for the company. So for those of the company, they want to do the phase 0 studies before they start formal phase 1, and just to reduce the risk of their investigation. Okay, then let's see the commonly used anti-cancer drugs already used in clinic. So here shows some of the commonly used anti-cancer drug types. The first one, as I mentioned before, is the cytotoxic drugs, which can cause toxicity in the cancer cells and then kill the cancer cells. Here shows the representative cytotoxic agents, including it's very commonly used in clinic, the cisplatin and the mitomycin and the methotrexate and the cytarabine and the paclitaxel. So all most of them target to nucleic acids. They can block the DNA synthesis or the DNA structure of the cancer cells, so that cause the toxicity of inside the cancer cells and then kill the cancer cells. And next is the hormone regulated drugs, which can regulate the hormone levels inside the patient, because some of the cancers, when they grow or proliferate, they need to high level of the hormone. So when using this kind of drugs, by reducing or sometimes increasing the hormone levels, they can control the growth of cancer, so that they can inhibit anti-cancer effects. This kind of drugs include toramycin and the mandosite progesterone. So these two drugs belongs to the hormone regulated drugs. And next is the molecular targeting therapy. This based on the recent studies of the oncology, some of the therapeutic targets have been found. For example, the EGFR, epidermal growth factor receptor, and the fibroblast growth factor receptor. These two receptors are highly expressed on cancer cells. So target to this receptor usually synthesize the inhibitor to block these receptors can exhibit anti-cancer effects. And also some other target for this PD-1 and PD-L1. We will introduce these two later in the following slide. And this HER2 is biomarker for the breast cancer. So targets to the HER2, for example, the monoclonal antibodies to block this HER2 can also treat the breast cancer. And last is the immunotherapy using some of the cytokines, for example, interferon and interleukin-2, which is the IL-2, and sometimes use the monoclonal antibodies to regulate their immune system level so they can promote strong immune response to kill the cancer cells. So this is 4 different commonly used types for cancer therapy, including the cytotoxic drugs, the hormone-regulated drugs, the targeted therapy, and the immunotherapy. And later, I would like to introduce them with more details. Then, it's one topic about the prevention of cancer. As we know, cancer is not easy to be defeated. It's difficult to cure them. So I think for most of the people, the prevention is more important than the therapy. If we can stop the formation of cancer, we will stay healthy. Once the cancer is developed, we need to pay a lot of money, and we need to eat a lot of. We need to suffer from a lot of therapy to kill the cancer. So the first choice is to prevention of the cancer. So how to do that? Because we have the immunosurveillance inside our body, our immune system working day and night to keep our safe to kill the cancer cells, right? So one idea is to improve the immune system to make we have a higher immune response to recognize the tumor cells to kill the cancer cells. Then how to do that? How to improve the immune system? First, we need to have a very healthy lifestyle. Here shows a picture. We eat healthy food. We eat enough vegetables. We eat fresh foods, and we do exercise. We drink a lot of water every day. We do exercise, and we sleep early at night. So all these healthy lifestyles can help to improve our immune system so that the immune system can protect us against tumor cells. And also another prevention choice is get vaccination. So why? As we mentioned, because more than 12% of the cancer development is related to the infection, right? So if we can get vaccination to avoid the infection, then we maybe can reduce the risk to develop cancer. So here, this is the HPV vaccine. If getting vaccine, they can reduce the cancer risk in a woman. And if unfortunately the cancer is developed, the next is the diagnosis of the cancer. And also body check every year is also important to find the cancer in the early stage. So if we keep every year go to the health check, and then we maybe can stop the cancer in the very beginning. And nowadays, tumor immunodiagnostic methods are commonly used because we already found a lot of biomarkers in cancer. And also these biomarkers has their specificity for the cancer type. So when we do the health check in the hospital, we get some blood, and then after the test, they can give you an idea. Do you have a high risk have cancer or not? Here shows the photos for some of the biomarkers. For example, for the liver cancer, the AFP and CEA, those are popular biomarkers for the detection of liver cancer. And also, the imaging technology help to find out tumor inside the patient. One technique which is very commonly used in China is the PET-CT. The full name for that is the Precision Emission Tomography Computed Tomography. Sorry, it's a long name. So we all usually call PET-CT. As you can see, using this PET-CT, we can see the location of the tumor, and also sometimes we can see the migration of the tumor. Because original, for example, in the lung, and then sometimes it can migrate to other organs. So use this imaging technique also can help the diagnosis of cancer. Sorry. And then if the patient developed cancer, and then next step would be their therapeutic options. They need to treat the cancer, right? So there's different options for the treatment of cancer. The first one, if the cancer have the sharpness of the border, it located only inside a small area, and then we can use surgery to remove the total cancer or the total tumor. And if usually after the surgery, the doctor will suggest use the radiotherapy also, because sometimes a little bit cancer cells may migrate to other tissues, but it's too small or too few to be monitored. So use this radiotherapy can kill those migrated cancer cells, just prevent the occurrence of their cancer back. So this radiotherapy is also used in clinic. If unfortunately the patient cancer already migrate to all over the body, then the surgeon cannot cut every tumor inside the patient. Otherwise, he will die, because tumors are and cancer cells are everywhere. So in this case, they can use only the chemotherapy, which means that they eat the anti-cancer drugs to kill the cancer. However, this chemotherapy has their limitation, including one, sometimes they cause side effects to normal cells. And also, sometimes these drugs may develop drug resistance in the therapy. And then maybe a better choice is the immunotherapy. And most of the scientists think that this immunotherapy will be the future to cure cancer. Here shows two scientists. One is the James Allison, another is Toshiko Honjo. So these two scientists get their Nobel Prize in Physiology or Medicine in 2018 because they discovered the cancer therapy, the mechanism, the negative inhibition of the immune regulation. We will introduce them here. So this one shows the immune system, how to kill the cancer cells. These cartoons help you to understand better. First, you can see the first one. This is your cancer cells. This gray cells is our cancer cells, and then we can remove or release the tumor antigen, which is labeled in red. The red bubbles are the tumor antigens released from these tumor cells, and then these antigens can be caught by the antigen presenting cells, for example, the macrophage or the dendritic cells. It can catch the tumor antigens. This can catch the tumor antigens, and then these antigen presenting cells can pass this antigen to the T cells to activate the T cells, and then these activated T cells can circulate in the vessels and then migrate to tumor tissues. You can see from the vessels to the tumor tissues. And then in the tumor tissues, these cancer cells can be recognized by the T cells. And then the T cells can kill the cancer cells. This is how the immune system works to kill the cancer cells. And then, as I mentioned, these two scientists find the mechanism, the negative control of this immunoregulation. So what they have found, you can see the first one is from the James Allison. He is an American immunologist, and he found one molecule which is important to control this immuno response to the cancer, the CTLA4. So this CTLA4 is a CD28 homolog with high affinity for B712. So what is the function of B712? I'm not sure if you have learned immunology or not. As you can see, this is a tumor-specific T cells, which usually plays an important role for killing the tumor cells. And then for in the bottom is the tumor cell or antigen presenting cells, the APC. Here they will have a B7 molecule which can bind to the CD28. And then for this binding, this antigen presenting cells can activate this tumor-specific T cells to kill the cancer cells. However, this CTLA4 looks similar to this CD28. So when the CTLA4 bind to the B7, then the T cells are silenced. They won't kill the cancer cells anymore because the B7 is broken. They cannot bind to CD28 anymore. So these T cells are inactivated, so that they will have a stop signal for the T cells. So they will see no cancer cells. They will think cancer cells may be good cells. They will not kill them anymore. So based on these findings, some new drugs are developed. For this one, this is the Yakwak. I don't know how to pronounce it. Its business name is Epinumab. So this is a monoclonal antibody blocking the CTLA4. Looks like this. This is a monoclonal antibody. It can bind to CTLA4 so that the CTLA4 cannot bind to B7 anymore, and then the CD28 can work normally to binding the B7, and then T cells can get activated to kill the cancer cells. So because this genes finds the CTLA4 and the mechanism to control the tumor toxicity of the T cells, so he get the Nobel Prize in 2018. And the next finding from this Japanese doctor, Toshiko Honjo, and what he found, he and his colleagues discovered a protein called programmed cell death protein 1, PD1. As you can see, this PD1 is located on the T cells, right? And in the tumor cells, they have the PDL1, which is the programmed cell death protein ligand 1. So this is the ligand. PDL1 is the ligand. PD1 is the receptor. So once they bind together, the tumor cells will give the signal to the T cells that, oh, I'm not abnormal cells, I'm a normal cells. So this PDL1 is a negative signal for the T cells. Once PD1 bind to the PDL1, and then T cells will not kill the tumor cells. So this is also a negative control signaling for the T cells. So based on Honjo's finding, we open a new way for the development of anti-PD1 cancer immunotherapy. Similar to the previous one, we can synthesize the monoclonal antibody to block the binding of PD1 to PDL1 so that the T cells will get activated and then kill the tumor cells. And next, new therapy for the cancer treatment is the cellular therapy. Here there's two different kinds. One is the CAR-T, the chimeric antibody receptor engineering T cells. And another one, TCRT, T cell receptor engineering T cells. So this one, I think it's easy to understand too. First, we have a cancer patient here, and here the red one means it's a tumor tissue. And then we can collect the tumor tissue, for example, do the tumor biopsy or do the surgery to cut tumor tissue. And then we can process to purify the cells. Because in the tumor tissue, we can collect the T cells. The T cells is the main cells to kill the cancer cells. So when we collect those tumor reactive T cells, when we culture these cells outside our body and then return to the patient, then this reactive T cells can help to kill all the other T cells, all the other cancer cells inside the patient. So this is one kind of cellular therapy. But sometimes if their immune system is too weak inside the patients, maybe they cannot collect enough number of T cells. Then we can do the modification of the T cells. Here, as you can see, this is a T cell receptor. And then on top of the T cell receptor, there is the domain to recognize the tumor antigen, right? So we can use the cloned technique to modify this T cell receptor so that this at the very beginning is a non-reactive T cells, maybe from some of the normal donors, and then modify them to teach them to recognize the tumor antigen, then culture in vitro, and then return back to the patient. So those normal T cells from other people after the modification, then feels back to the patient. They can also help to kill the cancer inside the patient. So at the top is the TCRT and the top is the CAR-T. So these two different techniques all belong to the cellular therapy for the cancer treatment. And last would be the gene editing technology. This is included one technique called CRISPR-Cas9. So what's CRISPR? The functions of CRISPR, the original name for this CRISPR is very long, is the clustered regularly interspaced short palindromic repeats. So this is some kind of thing found in bacteria, and this CRISPR associated genes, that's Cas. So CRISPR-Cas is complex look like this, which is labeled in blue here. So they are essential in adapting immunity in selected bacterial or any other organisms, enabling them to respond to eliminate invading genetic material. What does that mean? If you remember yesterday's class, I introduced the phage display technique, right? The phage. So the phage will infect the bacteria, and then they will invade their genetic materials inside the bacteria. So this CRISPR-Cas can help to remove those outside genetic material to keep the bacteria healthy. So this is a system for the bacteria to against the virus infection. So this system is found by these two women scientists. Also, these two ladies got the Nobel Prize last year, 2020. So what's the use for this CRISPR-Cas9? You can think it's just layer scissors, which can cut any gene you want. So for the application of CRISPR-Cas9 in cancer therapy, this shows one example. As I mentioned, because the T cell express PD-1, right? When the PD-1 on the T cells recognize the PD-L1 in the tumor cells, then the T cells will silence to the tumor cells. They won't kill tumor cells anymore. Then in this project, the doctors collect the T cells from the cancer patient and then culture the T cells in vitro, and then they use the CRISPR-Cas9 to cut the gene of PD-1 in the T cells, so that after the modification of this Cas9, then the T cells will not express PD-L1 anymore. So culture those T cells without PD-L1 and then culture them and fuse back to the patient. Those T cells would be reactive to the cancer, and then they can kill all the tumor cells inside the patient. So this is how to use the gene editing technology for the cancer therapy. So last, let's talk about the challenge of current cancer drugs. The first one, because the mechanism of the micro development is still unclear, sometimes if you use single drug for the treatment, usually the response would be relatively low, sometimes lower than 30%. So for most of the doctors, they will use the combined regimen to kill the cancer. And also because most of the anti-cancer drugs have a narrow toxic range, so the toxicity of the traditional cytotoxic drugs is high. So we need to maybe focus on more the molecularly targeting anti-cancer drugs development to reduce those side effects. And also sometimes after the long time treatment, these cancer drugs may induce drug resistance after the long-term administration. So we need to figure out the detailed mechanisms of the drug resistance so that we can solve this problem better in the future. So that's it for today's class. If you have any questions, welcome to send me emails anytime. Thank you so much. That's all for today's class. Okay, see you tomorrow. Bye-bye.
See you. Thank you.